Welcome. This video is to finish chapter 4, lecture 2. We're going to go over the different parts of the prokaryotic cell. So first we'll start with some general characteristics all prokaryotes have, and then we'll go into the different parts. So let's start by talking about some characteristics that all prokaryotes have. Now, all prokaryotic cells lack a nucleus. They don't have one, right? This is a defining feature. This is a way that we can tell prokaryotic cells apart from eukaryotic cells that do have a nucleus. Another thing that prokaryotes lack is membrane-bound organelles. You can think of a membrane-bound organelle like a compartment. Okay, in class we compared prokaryotic cells to a studio apartment and eukaryotic cells to a two-bedroom apartment. Okay, in a studio apartment, you don't have rooms. You walk through the door, you can see the kitchen, you can see the living room, you can see the bedroom all at once. Whereas in a two-bedroom apartment, the sleeping area is separated from the kitchen and the living room area, so you're going to have these different rooms. Okay, prokaryotes lack these different compartments, which means they lack different areas to do different types of chemistry. Now, this is actually important because this is actually going to go hand in hand with that surface area to volume ratio problem. Prokaryotes don't have the metabolism that's going to allow them to get bigger like eukaryotic cells. And the reason for that is the lack of these membrane-bound organelles or these different compartments or rooms to do different chemistry. So we need to keep that in mind. Prokaryotes are the open floor plan of cells. There's no division in them. There's a couple things else that we need to talk about when it comes to prokaryotes. It's been several lectures since we've talked about this, but we viewed a phylogenetic tree in class, or a family tree. Now, all life can be divided up into different groups. All life can be put into three domains, which are archaea, bacteria, and eukarya. Well, all three of those domains all come from a single common ancestor. We have a lot of evidence for this idea of how life started from a single cell. Prokaryotic cells are believed to be those first cells, that single common ancestor that all life sprang from. Now, both the domains archaea and bacteria, they're all prokaryotic cells, so they're all single-celled organisms. The eukarya domain, this is the eukaryotic cells. Okay, these can be single-celled eukaryotes or multicellular eukaryotes, just like me and you. Okay, but this is one of those things. Prokaryotic cells are thought to be the oldest type of cells there has been, and eukaryotic cells evolved from prokaryotic cells. Now, another thing that's unique to prokaryotic cells is that prokaryotic cells have a cell wall. Now, if you're thinking, Ms. Harker, they're not unique. Plants have cell walls. That's true. Plant cells are eukaryotic cells that do have a cell wall, but their cell walls are made from cellulose, right? That thing that we can't break down. Prokaryotes, on the other hand, have cell walls that are made from something called peptidoglycan. Peptidoglycan is this really interesting molecule that is only found in prokaryotes. It's not found anywhere else. Because of that, we can use peptidoglycan as what we call a drug target. Now, a lot of prokaryotes are bacteria. Some bacteria are pathogens, so they can cause disease. So if you get like a bacterial infection, the first thing the doctor is going to prescribe is an antibiotic. Most antibiotics are going to target the cell wall in bacteria. This means that antibiotics are specifically suited for disrupting this peptidoglycan layer, which is very important. If you ever go on to take micro, we talk a lot about peptidoglycan. Okay, so let's talk about some generalized structures in prokaryotes. Okay, now, to be fair, some prokaryotes don't have some of these structures, some have more than one of these structures, and some are going to be a combination of the two. Okay, now, one of our four things that all cells have in common are a cell or plasma membrane. All cells have this. Prokaryotic cells are no different. They do have this. Now, one of the things I want to point out here is where the cell membrane is. So if we follow my little pointer here, okay, the cell membrane is actually this inner layer. Okay, there's two layers that could be outside of the cell membrane in a prokaryotic cell. Now, the first one is going to be that cell wall. The cell wall is on the outside of the cell membrane. Okay, both the cell membrane and the cell wall are going to protect the cell and give the cell shape. Okay, they're also going to prevent the cell from drying out and dehydrating. Okay, so the cell membrane, just like we've talked about, is going to control what comes in and out of the cell, that exchange factor, that's going to give it its shape. And it's going to create that boundary that separates the inside of the cell from the outside. Now, cell walls also are going to help with this, but they're going to act as an outer covering that's going to protect the bacterial cell more than the cell membrane. In fact, a lot of times the cell wall is going to protect the cell from what is called osmotic pressure. So if we take a bacterial cell and throw it into a bunch of fresh water, what's going to happen is all that water is going to rush into the cell and cause it to burst. Now, the cell wall is going to prevent that from happening. Conversely, if we take the bacterial cell and we throw it into the desert where it's really, really dry, the cell wall is going to prevent it from completely drying out and collapsing in on itself. Okay, so both the cell membrane and cell wall have some pretty important functions. Now, as I said, we're going to talk about the cell membrane quite a bit. All of chapter 5 is about the cell membrane. It has a lot of jobs just besides being a boundary, regulating what comes in and out, and giving the cell shape. Okay, so those are our first two structures. Okay, so the next thing we're going to talk about are two more things that all cells have in common, right? All cells have DNA and all cells have ribosomes. Now, just because all cells have DNA doesn't mean we keep them in the same place. In prokaryotic cells, we're going to keep our DNA in what is called the nucleoid or the nucleoid region. Now, because prokaryotic cells don't have compartments, we don't have a special place to store our DNA like eukaryotic cells do. In fact, our DNA is going to kind of be free floating. It's going to be found mostly in the center of the cell. Now, prokaryotic DNA is really interesting because they only have one chromosome. We have 46. We have what are called linear or straight chromosomes, and they have what are called circular chromosomes. Okay, so prokaryotes are going to have one circle of DNA, and that's going to be kind of twisted together and folded up and looped around each other and held together by what are called DNA binding proteins to fit here in the center of that cell in that nucleoid region. Okay, and all organisms have more DNA than could normally fit in their cells without this twisting and looping over each other. Okay, so the genetic blueprint for all prokaryotic cells are going to be in this nucleoid region. Now, the other thing that we talked about that all cells have in common are ribosomes, right? Ribosomes are our protein makers. Okay, they're going to make proteins, and they're going to be found in the cytoplasm of the prokaryotic cells. So if we look here at our diagram, we have our nucleoid region here in the middle, and these little dots, these are going to be our ribosomes. They're going to be in our cytoplasm, which we probably should point out, if you ever were asked what this was, that in between all these ribosomes and nucleoid, that would be the cytoplasm, which is something else that all cells have in common. Okay, but one of the things that prokaryotic cells have that eukaryotic cells don't are they have things called plasmids. Now, plasmids are pretty cool. They're little circular DNA structures that carry one to a few genes. Okay, and they're not going to be actually part of the prokaryotic cell's genome. Okay, these plasmids can carry all sorts of genes. They could have genes that will help a bacterial cell survive when there's a drought. They could have genes that will help the cell be what we call antibiotic resistant. So plasmids are really cool because they are DNA that's not involved in reproduction, but they can confer different types of kind of superpowers to our prokaryotic cell. Plasmids are a really hot topic in biology. We use them all the time in biotechnology, but they're also a really big topic, especially in microbiology, because this is how we think we have so many bacteria becoming resistant to so many different types of antibiotics. Okay, now the outermost of some prokaryotic cells is sometimes what we call a capsule. Now this capsule is made of polysaccharides, like it's a sugar coating. Okay, the sugar coating is actually really important because it's going to protect both the cell membrane and the cell wall, and it's going to enable the cell to attach to different surfaces. It's sticky. Okay, it's also going to prevent the cell from being eaten by other cells. This is actually really important for some prokaryotic cells, especially if they gain access to a human host like us, because the first thing that's going to go after them are our macrophages or our white blood cells. Okay, it's their job to literally grab onto bacterial cells and eat them as fast as they can so they don't cause an infection. So this capsule is going to prevent our white blood cells from getting a good hold on the bacteria cells and getting them taken out of our system before they can cause an infection, which means that they can escape from our immune system. Okay. And just like our cell membrane, the capsule is going to keep the cell from drying out. It's going to keep it from dehydrating. It's going to help it keep its moisture. Okay, now what we're going to talk about on this slide are what we call the motility structures of prokaryotes, or how they move. Motility just means movement. Okay, so prokaryotes have two motility structures. They're called the pili and the flagella. Okay, they're going to use both of these to move around. Now, the flagella is the easiest thing to identify on any cell. Okay, so it's this long whip-like protrusion that is attached to the cell. These are going to, depending on what type of cell it is, it's going to either beat back and forth or it's going to rotate. Now, in bacteria and prokaryotes, they have a rotational pattern. So they're going to rotate. And the rotation of this flagella is what's going to move the bacteria along. Okay, bacteria need to move just like we do because they need to go from bad environments, like places with not enough nutrients, to good environments and places with lots of nutrients. Okay, flagella are going to be the main way that they're going to get there. The other motility structure is something called the pili. Now, the pili are these hair-like protrusions. Okay, we talked about these little hair-like structures when we talked about the surface area to volume ratio problem, because these protrusions are going to increase the surface area without increasing the volume of the cell. Okay, so these pili are hair-like structures. Now there's different sizes. The long ones are called pili, and the shorter ones are called fimbriae. Okay, so fimbriae are used just for attachment. This is how they're going to use the fimbriae to stick to surfaces. Okay, the pili are going to be like the flagella, and they're going to assist the bacterium in moving around. Okay, now we have special types of pili called conjugation pili. Now, conjugation pili are how bacteria cells can transfer plasmids back and forth. Okay, so they can literally grab hold of another bacteria cell and give it a plasmid. So this is one way that we can transfer things like the genes for antibiotic resistance, or the genes to form something called an endospore, which is going to allow bacteria to live in bad conditions without dying. Okay, so conjugation is the process of transferring this genetic material back and forth between cells. Okay, so that's it. That is the prokaryotic cell, and these are all the different things that you need to be able to label on the closed book portion of chapter 4. Okay, we need to know the pili and the flagella. I think those are the easiest ones because they are the motility structures. They're the ones on the outside of the cell. What's really hard is keeping the order of the capsule, cell wall, and cell membrane in order. The cell membrane is always on the interior. The cell wall is always on the outside of the cell membrane. For the most part, there's some prokaryotes that have two membranes, but we'll talk about those if you take microbiology. And then some bacteria are going to have these capsules. Now, not all bacteria have capsules, and not all bacteria have cell walls. In fact, not all bacteria have flagella. The E. coli that we grow in the back of the classroom for the microbiology class, because they live on petri dishes and they are given all the nutrients they could ever need, they have no reason to ever move. Okay, so they evolutionarily have lost their flagella because they don't need it. They don't need to move. Okay, ribosomes are the little dots in the center. Okay, the nucleoid regions where our chromosome or our DNA is. And those are all the structures that you need to know. Probably the easiest diagram to label for cells is the prokaryotic cell because they do have a limited number of structures that make them up. Okay, we'll see you in lecture 3.